things that happen within the cell. The cell is something that it's not very easy to explain. You all have been to high school and you should know what is a cell, but there is some details that it's possibly worth reminding. Cells are actually small structures. They go from 5 to 50 microns. Microns are meters by 10 to the minus 6. So 1 million of a meter. That is 1,000th of a millimeter. In 1 millimeter, you can have 100, 500 cells or so on. I'm not going to go through all of these dimensions. I will just mention, for example, that the DNA, the double helix of the DNA that we all think we know, is like 2 nanometers wide. Now, to actually realize the dimensions that we see here, we exploded a cell to 10 million times. That means that a cell which is 5 to 50 micron, we should consider as large as a village, like 50 to 500 meters wide. The only difference we must keep in mind is that the cell is actually in 3D. So it's a volume, whereas a village is usually a flat or a very, well, the one dimension is actually very small relative to the others. In this context, the DNA in a human cell would be the dimension of a pipe, like a water pipe of 2 centimeters wide, and its length would be 20,000 kilometers. That is like the distance between the North to the South Pole, passing along the surface of the planet, not through. All this DNA has to be wrapped within the nucleus of a cell, and it has to be wrapped in a way that it is accessible at all times. So it can't be all wrapped around like in a wall ball. So it has to be accessible all the time. Again, another minor thing I want you to notice is the dimension of a virus. Viruses are very small relative to cells. And again, in our 10 million times explosion, a virus would be the size of a small human. As you can imagine, one single virus does almost nothing to a cell. To have an effect, you need at least 100. If it's very bad, like a large squad of really nasty kids, or 1,000 to 10,000. Just to give you an idea of the dimension and of the things that go on in cells. We are mostly interested in proteins, and we use Blender to move and to visualize proteins. First of all, we have some problems that come from the science of proteins that we have to solve, and problems are, first, that many times data relative to proteins are incomplete. That's because the understanding of proteins has to be done through quite sophisticated techniques that require that proteins are still. So moving proteins cannot be visualized. The way to get information about the shape and the way proteins are is through X-ray that requires the proteins are fixed and crystallized. Because many proteins in nature actually like to move, an easy way to have them still is to remove the parts that drive the motion. So if we go back to a metaphor of 10 million times, we can imagine that to study protein, we do some kind of experiment. You could think about, for example, some people or some beings from an extraterrestrial planet like that are willing to study objects on Earth. Say, for example, they want to study cats. They can't see cats because they are too small for them to see. So what they do, they just try to remove the parts that move the most, have a number of cats all fixed, and then they can actually make a picture of the cat. Now, it's not quite a real cat, but it does give you a lot of information. It gives you an idea of the general shape. It tells you how the bones are placed, and the cat is actually still alive. It can breathe, it can eat, and do all of its functions. And the Martians, they know that something is missing, so they can try to replace the missing parts by analogy with some other parts that maybe they have studied in another situation. So, for example, because cats belong to the class of vertebrates, they can imagine that the appendices that they have removed might look like some of the appendices that they have already seen in other animals. Eventually, however, some animals more similar to cats can be visualized even by Martian, and they can understand that cats are not just vertebrates, but they are also mammals. So they put appendices more like to the ones that we see mammals. Not everything is always perfectly right, but it's one way to get an idea of how cats work on planet. Now, as I said, we don't actually have cats, we have proteins. And proteins don't work like animals. They are a little bit the different objects. I will skip this and come back to that one later. Proteins are made of atoms, you have seen before, and they are actually described as collection of atoms, and the description is the 3D coordinates of each of the atoms that is composing the proteins. The small ones can go just on several hundreds to a few thousand atoms. Most complex proteins can get to the many, many thousands of atoms. Another information that we have is the way that these atoms are linked to one another. The link that connects each atom to one another are fixed. They do not change. So like the cat, the front legs are always in the front, the back leg always in the back, the tail stays where it is, but, and still they are flexible, they can move. And the way that atoms move is by rotation along the axis that connects each two of them. Well, I will just spare you the description of the side chains of proteins, but I will just tell you that there is one main chain. So there is a chain which is a very, very long chain of single units, which are characterized by the specificity. So we have one unit, NC alpha C, another NC alpha C, another NC alpha C, and to the C alpha is attached another unit which is different for every one of these units. Here we have an example. We see that the main chain, so this NC alpha C chain, is moving by rotation around these two dihedral angles or torsion angles. And the side chains, of which we can see here a very small one, again, move by rotation of this angle. So for example, if we want to move, well, if we see that in one position, this atom is here, and in another position, this atom goes here, we should think that, first of all, there's been a rotation around this angle, which is key 3. So it goes 180 degrees to go here. And then there is a rotation around this one that will just move the entire chain from here, from this hinge of whichever 30, 40 degrees to go here. In this case, this angle is not rotating. So we know the position, and we know that to move from one position to another, the only thing that can move is the rotation around connecting joints or bones, or we call them bonds in chemical terms. the length of the bond and the planar angle that are defined by each 3 atoms are usually not changing, or if they change very, very little. So we move the atoms because we want to see the proteins moving. Now, the information on motion is also quite difficult to get. Again, we go back to the cat, and we see that the scientists on this planet have taken pictures of the cat, and each one take a picture in a different conformation, in a different position. We call them conformations when we talk about proteins. So from this collection of data, we want to get to a meaningful movement for the cat or the protein. So the first thing we have to do is to align them. Now, it's very easy to see that this, if we flip it and turn it, we can align these 2 cats quite easily. This one can also be aligned, not very difficult. And this one also, it can be acceptable. And we can try to interpolate between all these conformations to see the movement of the cat. But then maybe some other scientist comes up and said, OK, I have seen a cat, and it's completely different. It has a roundish shape. The front end and the tail end are actually connected to one another. And the relation with the surface is done by the part of the body and not just by the tip of the extremities. So then some dispute can start between scientists, and of course they are all right. We are trying to put them to find an agreement between them, saying that the cat can go from this conformation to this conformation. It is very difficult, of course. For small motions, like the ones connecting 2 similar conformations, the game engine of Blender is good enough, is good enough to get a number of intermediate conformations that can be acceptable from a physical and chemical point of view. I'm not going to describe in the details of the minimization cycles or of the distance, the RMSD, but it is good enough. We can get intermediate conformations that are actually sensible, can be accepted. However, for very large changes in conformation, like say from the sleeping cat to the jumping cat or to the standing cat, then we have to invoke some more complicated system. So I'm going to show you this because it is actually a work in progress, and we are open to suggestion if any of you has a good idea. We have done this from the coordinates. We have transformed all the coordinates information into rotation angles, the rotation angles that connect all the atoms. The difference between the rotation angle in one conformation and another conformation, it can also be expressed in a rotation. So we have a collection of quaternions, each assigned to every bond, which basically means to every atom. Now, we have to set a system that puts together all these changes in conformation, taking into account mostly the collisions, which is quite complex, and sometimes also the other forces like electric forces and lipophilic forces that apply to molecules. So we are thinking of how to solve this problem, and one possibility is the calculation, to set up a learning system that tries to get directly from one conformation to the other, and every time that finds an obstacle, learns that there is an obstacle there and can start again, setting an intermediate aim before getting to the final aim. Now, this is not a very difficult problem for 2- or 3-dimensional space, but here we have an n-dimensional space where n is the number of atoms, which goes to the many thousands. So it could be quite difficult. Another possibility is that we start the system going through a direct interpolation, direct quaternion interpolation, of course, because it's always rotation. And whenever there is an obstacle or some reason to stop, the atoms that are directly colliding should stop the rotation, and the stop should propagate back from the side chain to the main chain. And when it reaches the main chain, it should propagate in one or the other direction or in both directions until it finds a movement that allows the system to evolve towards the final aim without actually provoking collisions. If this doesn't work, then another possibility is that if we have a system that is colliding, we want to go from here to here, and there is a collision in between. If this rotation cannot be done at all, then we should consider the possibility of doing the complementary rotation so that we reach the final conformation without getting into collision problems. So this is a work in progress, and we hope we can relate to you some better information for next year. So much for the movement. The second subject that we work on and that we use Blender is the visualization. We don't want to see atoms. In the academic field, most of the times proteins and other biological molecules are represented as atoms. Sometimes they have bonds in between, sometimes not. Most of the times they do have bonds, which is, it does provide quite a good information. It's an extremely fine description. And of course, because it's done primarily by the physicists and chemists, it has quite a chemical perspective, which can be very informative. However, in biology, sometimes it's more important to see the properties that are due to the interaction of many atoms or of large parts of a protein, and that cannot be perceived if we watch single atoms. So we decided to, we actually elaborate a visual code to show proteins in a different way. This visual code includes the calculation of the surface, so that means the space occupied by the atoms. Actually, this is defined as the space which is not accessible from the water to the water that surrounds the protein. It's called surface accessible, solvent accessible surface area, that's SA. On this surface, we want to map the lipophilic and the electric potential. These potentials are, again, calculated with external programs, and we want to map them on the surface. I think Mike will enter some details of this, so I will not I will not say any more about it. These are some of the ways that are used to represent proteins. As you see, you have seen before some of these ribbons. Just a few minutes ago, there was a very nice representation in Blender with the ribbons shiny and looking solid. here we have bonds, here we have atoms as points, and here we have atoms as their van der Waals radius. Okay. Starting, I don't know where I'm going. Okay. So this is one of the proteins we have. Here we have all the atoms composing the protein. They are about 2,000 to 300. We calculate the surface. On the surface, we can map the lipophilic potential, which is calculated externally, and is introduced in Blender as a value from 0 to 1, which can be very easily visualized with shades of gray. However, shades of gray are not good enough for us because we want to give a realistic and intuitive representation of the physical property. So we have devised this, visualization system that assigns a shiny surface to the more lipophilic areas that are the ones that like to react with other proteins, and a rough and dull surface to the parts that are more likely to stay free in water. Here we have another image, a very fine rendering of calmodulin. I think this is actually demonstrating how you can be tricked to think that this object is actually existing, and you can attribute intuitively and visually properties to the surface of this molecule. Okay, so this, of course, I did almost nothing of it. It's been done by everyone else in here except me. This is basically our laboratory. We have mathematicians, physicists, chemists, artists, another biologist, programmers, and another mathematician. And we have Mike. Actually, Mike is not anymore in the lab, but I don't want to take him off because I hope he will keep working with us in the future, and I let him speak now. Thank you.
So what does this have to... Can we switch to this? Thank you. What does this have to do with Blender? Well, basically, we're using the Blender for three things. When we started this project, the research on what I'm calling protein expression, the start of the movie, as well as the research on motions, it was we tried some other tools. We were using a lot of the scientific tools as well, but in the end, Blender was chosen as the visualization platform. So in 2009, this is when I joined the team and started working mostly with Blender 2.5 and trying to integrate all the previous research into basically one interface under Blender 2.5. And the idea is that we'll have something so that the regular users, someone who is not training Blender, for example, be able to use this software and produce stunning-looking animations while still doing real science. So, and I think in 2010, this year we'll still continue working on the protein motions, as Monica already said. So what exactly is BioBlender? It's an extension of Blender 2.5 that allows researchers to import and view protein models. Protein models, as we also described, they are usually stored as PDB files. It's a text file that describes the coordinates of each atom. So it's we are able to import that into Blender and basically view them in any way the scientist want. So this is what this program looks like. I changed the splash screen as well. But basically, you have this interface which we sort of adapted to the workflow of a scientist, of a researcher. And so it's like this. To the left, you have mostly a panel where all the actions would happen, and on the right, you have a 3D view. Of course, you can change the views. So on the right, you can rotate the views. You can basically do everything you can do in Blender. So BioBlender, as we mentioned, can do primarily 3 things right now. It's able to import molecules into Blender, into itself. So it reads the data files and generate a 3D model based on that description. Secondly, it takes that model, it takes that 3D model and applies the Blender physics engine, the physics engine in the game engine to simulate the collisions and the dynamics of it. So when you import the model, it's able to resolve the collisions that's a result of a protein motion. And thirdly, we added an interface that basically allows an artist to more easily create visualizations based on some of the techniques that we've been developing. So I'll cover more cover more about that later. Basically, BioBlender is a result of having to deal with all kind of different scripts and batch files across different operating systems. And it was really not a very nice workflow. So we decided that we want to bring everything into one interface, make it easier to manage, make it easier to use for, well, everyone. Why do we want to visualize? What's wrong with this picture? Can anyone see what it is? Basically, we want to turn that into something that someone who works in the field can easily recognize. More technically, BioBlender is a collection of Python scripts that runs within Blender 2.5. It's loaded as an add-on, and because we are still limited to how much science we can do from within Python, some of the more specialized features are called as third-party plugins. So we do have some binaries. Well, most of them are free that the BioBlender application will use. So as I mentioned, right now BioBlender can do 3 things. It can visualize the molecules, run the physics simulation, as well as generating movies with really good visual quality. So because we can do 3 things and we don't really know what the researchers want, we have to make sure the interface is set up in a way that's intuitive to them and not slowing them down while they figure out what all the buttons do. So BioBlender is integrated into Blender with this engine selector here. So the new engine selector lets you hide some of the features that you don't want while exposing new features that you do want. For example, if you select the game engine, it will hide all of the features that does not apply in the game engine, and vice versa. So when you select the BioBlender engine, it's right now it doesn't hide so much. It doesn't hide anything else, but it exposes all the BioBlender features that one can expect. So first thing, I'm going to talk about the molecular visualization. So when we import the molecules and visualize them in 3D. This is a bit technical, but basically it reads, this is all Python. So the Python script reads the PDB file, which is a protein it's a description of a protein. It creates the 3D objects, and if the user wants, it will also create the physics constraints using the Blender physics system to create the constraints, which limits the movement of each atom on specific axis. So the benefit of this is that when we run the physics simulation, it will be able to only pivot around some of the axes. And because sometimes protein comes in multiple positions, conformations, if we load multiple models into the same scene, it's able to create a keyframe automatically, so the protein would animate from one position to the other. And once the model is loaded, you can pretty much it would exist in Blender as a 3D model, so you can do most of the typical things you can do with a model. You can rotate around, view it from different angles. And in fact, you can go into the game engine to simulate the physics. So this is sort of what it looks like. This is a rendered output, so it looks slightly better than the quality from the viewport. But I will do a live demonstration of this after I finish the talk. The second part is basically running everything inside the game engine. As I said, you would load everything. You would load the molecules that has different conformations into Blender and basically apply the physics engine to and then to see if they collide.